Is everybody hearing me okay? We good? Can you hear me back in the nosebleed section? We good? Alright. Okay, well, good morning, everybody. It's a pleasure to be here again for the military tropical medicine course. So this morning, well, today, I guess in general, is really Wounds Day, and you already heard about nematodes from Dr. Mitri, so we're going to continue in that vein, but with a completely different group of worms for the next 2 hours. And all the worms that we'll be discussing in the next 2 hours are trematodes. So just a little bit of introduction as to what these organisms are. So trematodes are an example of a parasitic flatworm. And there are two main groups of well-known parasites that are parasitic flatworms. The trematodes are one. The other are the cestodes or tapeworms, and I understand you'll be hearing about them in great detail from other lecturers, probably from Dr. Nash from NIH principally. But these two groups, the parasitic flatworms, they're quite distinct from the parasitic roundworms or nematodes that Dr. Mitri was talking about. So their biology and their life cycles and the way they cause disease are also quite distinct from what you've heard already this morning. So, as far as trematodes or flukes are concerned, there are actually many different species that are capable of infecting humans and causing disease. And in fact, over 60 different species of trematodes have been identified as causing infections in humans. And obviously this morning we have no hope of doing anything but just raising the surface of that. But I will focus on those species which are the principal pathogens, the most important pathogens in terms of their potential to cause disease and also their prevalence around the world. Now, in the first hour, we'll focus on a group of basically 5 different trematodes, 5 different genera, these species over here. And in the second hour, we have a separate hour reserved to discuss the schistosomes. Now, the reason for this is because the schistosomes are arguably considerably more pathogenic than these other organisms. And they also, just numerically, infect way more people than the rest of these other organisms combined. In fact, the schistosomes are some of the most important parasite infections of humans worldwide. So we'll return to discuss the schistosomes in the second hour this morning. And in the first hour, we'll focus on these 5 over here. So I apologize for the Latin names, but that's just how it is. But to sort of break this down and make it more digestible, the first 3 genera here are what we call liver flukes, because they principally cause disease in the liver. Then this 4th one down here, Paragonymous, this is the lung fluke, which causes disease in, guess where, the lung. And then finally, we'll end up with a discussion of Fasciola, the intestinal fluke, which lives in the intestine. Actually, it's not very pathogenic at all, but is quite widespread in certain parts of the world. Yeah, so these guys are named then based on where they end up in the body and where they end up causing disease. In terms of numbers, it's thought that around 20 million people worldwide are infected with liver flukes, another 20-odd million infected with lung flukes, and an unknown number infected with Fasciolopsis, but it could be tens of millions, maybe even 100 million or more. And so these are not insignificant. So these are very prevalent pathogens on a global level. Contrast that with the schistosomes, which collectively infect probably 250 million people worldwide, maybe even more. Okay, so we'll come back to discuss those in the next hour. So a couple of sort of introductory remarks then about trematodes, features that they all share. These are typical flukes. They have a flat, leaf-like sort of body form. They can vary greatly in size depending on species. The smallest ones are just a couple of millimeters long, whereas some of the bigger ones, as I'll show you, are quite impressive organisms, can be several inches in length. Okay, and we'll have some specimens of these organisms there for you to look at in the lab this afternoon. They all have two suckers. They're quite striking looking organisms. Two suckers at the anterior end, one that surrounds the mouth, and then another on the underside of the body that helps them to attach to host tissues. And then being flatworms, they have a blind-ended gut. They have a mouth that leads to a short pharynx that then leads to a blind-ended gut that usually runs the length of the body. And so anything that is one of the downsides of being a flatworm is that anything that you eat goes in through your mouth has to come back out through that same orifice. And then the vast majority of trematodes are hermaphrodite, meaning that each individual organism has a functional set of both male and female reproductive organs. Okay? There are some exceptions, the schistosomes being one of them. So we'll highlight that difference in biology when we come back to talk about that later. So this is just a generalized sort of diagram of what a fluke looks like. I mentioned the two suckers at the anterior end. There's the oral sucker and the ventral sucker. And then behind the oral sucker, there's this short pharynx, and then this quickly divides to form these two ceca of the gut that run the length of the body. And then basically the rest of the insides of these organisms are stuffed full of the various reproductive organs, both male and female. The ovary is usually located centrally and connected to a uterus that stores eggs prior to their release. And then the testes are usually located ventrally. And then the rest of these glandular structures around the periphery, these are what we call vitellaria or vitelline glands, which produce substances that contribute to the shell of the egg, to the formation of those eggs. So you don't need to worry about all these various details, but they're often quite easy to see on specimens that have been stained appropriately. An example of some such specimens here. So you can see immediately there's tremendous variation in size. Some of the smallest organisms are tiny through to Fasciolopsis, the intestinal fluke, which is one of the largest parasites of humans, can be several inches in length. So there's also some commonalities to their life cycles too. So another aspect of trematode biology is that all of these organisms all require a molluscan intermediate host as part of their life cycle. So in addition to the human or mammalian host, in order for the life cycle to complete, they all require a snail intermediate host. And all the species I'm talking about this morning, these are also zoonoses, meaning that in addition to humans, they can also utilize other definitive mammalian hosts. And this poses a serious problem then from an epidemiological and control perspective, where all these other host species can act as a reservoir of infection for humans. Another interesting aspect of the parasites that I'm discussing in this first era is that they are all essentially foodborne illnesses, if you like, in that infection requires that you ingest the infectious larval stage. And this is not the case with the schistosomes. Again, we'll come back to focus on that difference in a second. All the organisms I'm talking about in this first era are essentially foodborne illnesses. And they're transmitted in different ways, and as we discussed the different life cycles, you've seen how they differ from species to species. But a common feature of all of them is it requires that you ingest something in order to get infected. All right, and so this is a generalized sort of trematode life cycle that I threw together with my fantastic graphical skills in PowerPoint. Okay, so you have the definitive mammalian host over here, and then that obligatory snail intermediate host. And the snail intermediate host becomes infected from larval stages that hatch from eggs that pass out of the human host. And then there's a process of development that occurs in the snail that results in the production of another larval stage called a cercaria. And then before this can become infectious now for the human host, so that the life cycle can be completed, the cercaria has to transition into a sort of a dormant, resting larval stage called metacercaria. And it's this stage that's actually infectious for humans and must be ingested in order for infection to occur. And this transformation to the metacercaria can occur in a variety of different places depending on the species that we're talking about. So some species utilize a second intermediate host to host the metacercaria. And so we'll look at examples of this. Some species utilize other vertebrates like fish. So contaminated fish becomes a source of infection. Other species utilize other organisms like crustaceans. So infected crustaceans can be a source of infection. And yet other species actually insist directly on the leaves of vegetation. So they're eaten along with the leaves and other components of these various plants that are foodstuffs for humans. Alright, so let's start then with a discussion of the liver flukes. And I showed you that there were 3 different genera of liver flukes that we needed to be that I needed to be aware of. The first 2, the good news is that they are so similar to each other that we can discuss them together. Everything I have to say about one is equally applicable to the other. So in the case of these 2 genera, opithorchus and Clonorchis, these are quite closely related, and we can basically discuss discuss them as one. Their life cycle is the same, the way they cause disease is the same, and so on. Okay, so a couple of pictures of these organisms to give you a sense of what they look like. These are about a centimeter or a little more in length. No need to worry about the details, but these are stained examples that show you all those various reproductive structures inside. They can vary quite a bit in shape. They're usually sort of a lanceolate sort of shape. Sometimes they can look a little chubbier, and they have those suckers at the anterior. Now, this is Clonorchis, Clonorchis sinensis. Okay, you can tell by its common name where in the world this is found. It's known as the Chinese little giant ant. I'll show you a map in a minute. Then there are 2 species of opithorchus. The first one, opithorchus vivarini, is found in Southeast Asia, and so it's known as the Southeast Asian liver fluke. Well, a lot of imagination went into this name, but basically looks identical to Clonorchis. And then there's a second species of opithorchus, opithorchus phailinius, also known as the cat liver fluke, because another important host of this parasite are various species of cat. And it, again, looks very similar to the other 2 species. Okay? So here's a map then of where these parasites occur in the world. So a heavily Asia-centric distribution. Clonorchis almost exclusively found in China, as the name suggests, but also in a few other countries in East and Southeast Asia, such as in Korea. Opithorchus vivarini, in the dark color there, in Southeast Asia, in Thailand and Laos and other nearby areas. And then Opithorchus phailinius has a more westerly distribution from Central Asia westward all the way to as far as Eastern Europe, in fact. Okay, so here's the more detailed Opithorchus Clonorchis specific life cycle from the CDC. So the human host over here, and the adult flukes live within the lumen of the bile duct, actually within the smaller intrahepatic portions of the biliary trees, high up in the biliary tree. And in that location, they produce eggs which pass down the bile duct to the duodenum and then leave the body in feces. And so the eggs get back into the environment in human feces. When they reach fresh water, where the appropriate snail intermediate host is living, snails actually feed on the eggs. They ingest the eggs, and the eggs hatch inside the snail. This is how the snail becomes infected. So there's several rounds of development that occur in the snail that you don't need to worry about, but it culminates with the development of this life cycle stage called the cercaria, which is then released from the snail and exists briefly as a sort of free-living aquatic stage. And the job of the cercaria is to locate the preferred second intermediate host, which in the case of these parasites are various species of freshwater fish. So the cercaria will invade the fish tissues and insist in the tissues of the fish and become the metacercaria. And the definitive host becomes infected when fish tissue containing metacercaria is ingested. So eating to infection with this parasite is associated with eating either raw fish or fish that hasn't been cooked, maybe prepared in some other way, such as pickling or smoking, something which doesn't inactivate the metacercaria. If the fish was cooked, it's completely safe. The metacercaria is killed. But if it's eaten in some other way that doesn't involve cooking, then the metacercaria survives, it passes through the stomach, and on reaching the small intestine, it now reactivates, it sort of wakes up, comes back to life. And what emerges from the metacercaria is a juvenile fluke. And then something like completely remarkable happens. The fluke actually finds the opening to the bile duct in the small intestine, and it crawls up the bile duct into the upper portions of the biliary tree, and that's where it grows and develops into an adult liver. Okay? And then the life cycle is completed. Any questions about the liver fluke, the life cycle before we move on? Right. Okay, so how do these organisms actually cause disease then? Well, it's the presence of the adult flukes in those intrahepatic portions of the bile duct that elicit an inflammatory reaction and ultimately lead to disease. So over here we have a normal bile duct in an uninfected liver. So you see that standard cuboidal epithelium of the bile duct here, and healthy hepatocytes either side. Over here we've got a duct that has a fluke in it. So this thing in the middle here is the fluke. In fact, you can see the two ceca of the blind end gut in cross section as well. And you notice that there's some significant changes that have occurred here. So the bile duct epithelium is no longer a single layer of cuboidal cells, as it should be. You can see that it's actually several cells thick in some places now. And in fact, there's evidence of cell division with some mitotic figures in here. So the bile duct epithelium is proliferating in response to the presence of a parasite. And then there's also this accumulation of inflammatory cells in the tissues immediately surrounding the bile duct. There are lymphocytes, macrophages, eosinophils, and so on, accumulating in the tissues around the bile duct. So in more chronic lesions, so these flukes can live for many months, perhaps even years. And so there's this chronic inflammation of the bile duct epithelium and the surrounding tissue. And ultimately, this progresses to fibrosis. As you can see over here now, there's the appearance of fibrotic tissue as a component of this inflammatory reaction. And over here, an even more established lesion, we now have this thick layer of fibrosis surrounding the bile duct. We can visualize these changes in various ways. I like this picture. You wouldn't sort of normally do this, but this is a contrast cholangiogram. And you can see immediately how contorted and twisted and irregular the bile ducts have become. You notice that these little arrows are pointing to what appear to be little filling defects. in the contrast medium. So each, at the tip of each of these arrows, these filling defects, these are the actual flukes. So they're large enough to exclude the contrast medium, so you can actually see them, visualize them directly by this method. So what does disease caused by these flukes look like? So typically the presentation is that of a chronic disease. And in fact, I should say that these organisms are not especially pathogenic in and of themselves. And disease usually only manifests itself when somebody has a relatively heavy infection. And then it usually takes many months to develop. So the typical presentation is that of a chronic disease that develops over many months, characterized by fatigue, abdominal pain, sort of vague symptoms, some dyspepsia, and maybe obvious enlargement of the liver. In very rare circumstances, if somebody was infected with a large number of metasaccaridia all at once, so you binged on raw fish or something in one entire meal, it can result in an acute presentation with sudden onset of fever and abdominal pain and so on. That is how we know. Now there is a more sinister aspect to this disease, actually a very important one that I want to emphasize, and that is associated with this stimulation of bile duct proliferation that occurs. So this can ultimately lead to changes in the bile duct, to metaplasia of the bile duct epithelial cells, and ultimately to full-blown neoplastic transformation of those cells, and then the occurrence of cholangiocarcinoma. And this is a section here of such a lesion. You can see these dense swirls of these cuboidal-like cells derived from the original bile duct epithelial cells. Now, in most of the world, this is a very rare liver tumor, right? Hepatocellular carcinoma is by far the most common liver tumor in most of the world. But in areas where these liver flukes occur, the incidence of those two cancers is flipped because of the oncogenic potential of this parasite. And unfortunately, this is a cancer that has quite a poor prognosis. And so for this reason, these liver flukes are classified by the WHO as group 1 carcinogens because of that tremendous oncogenic potential that they have. The only other eukaryotic pathogen that has that distinction is one of the schistosomes that we'll talk about in the second arrow this morning. So here's a graph just illustrating how radically different the breakdown of liver cancers is in parts of the world where these liver flukes are found. So in most regions, liver cancer is not that common, and hepatocellular carcinoma tends to predominate. But in places like Thailand, so that the incidence of liver cancer is much higher, and the cholangiocarcinoma is much, much higher. In fact, this map, this is from a colleague of mine at George Washington, who studies these infections at various sites in northeastern Thailand, and this map quite clearly illustrates how well the incidence of cholangiocarcinoma overlaps with the prevalence of opithorchus viverini infection. Okay. All right. So how is this diagnosed? Well, typically it's by demonstrating the presence of eggs in stool. You can treat an individual with an anthelmintic that kills the flukes, and then recover the flukes from the stool, but you have to sort of already have an inkling that that's the infection that they have in order to do that. And then ultrasound of the bile ducts usually reveals a fairly characteristic pattern, so I'm told anyway, by people with expertise in ultrasonography. Okay. So what do the eggs look like? We'll have examples of these for you to look at in the lab this afternoon. But typically the eggs of all three of these species are actually some of the smallest eggs, helminth eggs that you find in human stool, only up to around 35 microns in length. They have an interesting structure in that they have what's called an operculum at one end, which is essentially a little escape hatch that pops open to allow the larva inside to emerge once the egg has been eaten by the snail intermediate host. And then they have some other features as well, such as a little bump at the other end of the eggshell called the opithorchus knob. And hopefully we'll have some examples of these. We can discuss this more in lab this afternoon. And then the eggs of opithorchus essentially identical. Okay? In fact, we don't really the eggs of these two genera, of all three of these species, are so similar that we don't distinguish between them just by visual observation. Okay. And then the treatment, standard treatment for this disease is a dose of several doses of praziquantel. This is a broad spectrum anthelmintic with activity against a wide variety of different flatworms, both tapeworms and flukes. It's a recommended treatment for most flatworm infections. There are at least one exception to that rule, which I'll point out when we get to it. Okay, so here's a couple of pictures then of just parts of the world where these infections are prevalent. So these are some images from a colleague of mine from China. And in China and other parts of East Asia and Southeast Asia, fish are a very important part of the diet. And often these are farmed, these fish are farmed in these aquaculture operations. These impoundments are created and flooded, and the fish are raised in these ponds. And many of the fish that act as a source of infection are farmed in this way. In fact, over 100 different species have been found to serve as a second intermediate host for the Metacicaria. One thing they have in common, though, is that they're all member of the carp family, the Cyprinidae. So this is the same family of fish that include the koi carp and so on, ornamental fish that are not usually eaten, but a wide variety of other species in that family are often eaten in East Asia. So these fish can look quite different. They're often either eaten raw, or perhaps salted, or fermented, or smoked, but in some way that doesn't inactivate the Metacicaria, and therefore these fish remain a hazard when they arrive on the dinner table. Now, another common practice in these parts of the world is that human waste, feces and urine, is often used to enrich the waters in these ponds. They're considered as a source of nutrients, and so these are allowed to enter these ponds to enrich the water. So that's what's happening here. This pipe is carrying human waste into this pond. You can see the fish all gathering here, all excited for what was going to pop out at the end of this tube. But remember, it's the snails down in here as well that's the real problem. Those are going to eat the eggs that are in those in the feces, and then become a source of infection for the fish. So you have everything that you need for completion of the life cycle all going on in this pond. Okay? And another example of human waste being introduced into one of these aquaculture ponds. And then to my colleague, Dr. Brindley, he says that particularly in places like northeastern Thailand and the Lao Republic, there's a dish called koi pla, which from what I can understand is, perhaps some of you are familiar with this and know way more about this than I do. But it sounds like it's almost like a kind of a ceviche. Like fish is chopped very finely and mixed with various herbs and spices and so on. So it's quite spicy. This is a very popular dish in this part of the world. And so the fish isn't cooked, however, in the preparation of this dish. And so it becomes a source of infection for those who partake. Right. Any questions about those liver fluids before we get started? Yes?
So what do you think the infection rate of the fish farms are? Like 100% probably?
Yeah, I think if you go back to that map that I showed you, I don't know about the fish farmers themselves, but in those communities in general, the prevalence of infection can be very high, 30, 40, 50%. All right, so yes, so
Is there, do you ever see symptoms of acute obstruction?
that's a very good question. So, not typically with these foods, but with these next ones I'm going to talk about, the answer is yes. Okay, so hold on to that thought. Any other Any other questions for Christine? All right. So there's another group of liver flukes that we need to talk about. I'm going to discuss these separately because there are some important differences in their biology and the way that they cause disease that I want to highlight. So there's another genus of liver flukes called Fasciola. It belongs to a different family. Okay? So biologically quite distinct from liver flukes we've talked about already. Now, back to your question. This is actually a much bigger organism. Okay, so there are actually two species of Fasciola. The common one, the most widespread one around the world, is Fasciola hepatica, and it can be an inch long. Okay, and then the second species, Fasciola gigantica, the name tells you this is a really substantial organism. It can be over twice as big as Fasciola hepatica, so 2 inches or more in length. So these are liver flukes that, again, live in the bile ducts like the flukes that we just talked about, that are substantially bigger. And so the potential for complete obstruction of the bile duct is a much more likely occurrence. So where are these organisms found? Well, this is a general distribution of both species. You'll notice it's found throughout the world, right? Even in North America and throughout Europe, all through South America. This is not a tropical, well, it's found in the tropics, but it's not restricted to the tropics. It's found all over. So why don't we hear more about this? Why don't people in the US infected with this? Well, this is sort of more of a kind of a horrible map, but it's broken down by the two species. So most of that distribution worldwide is Fasciola hepatica. Fasciola gigantica typically is what's found in sub-Saharan Africa and over in the Indian subcontinent. All right? Right. So Fasciola gigantica, strictly speaking, really is a tropical parasite, whereas hepatica is found basically throughout the world. So this is really a parasite of livestock, wild animals and domestic livestock. And humans are really an accidental host for this species. And this parasite is still a major problem for farmers in the US and throughout Europe. For sheep farmers, for cattle ranchers, and so on. This parasite is still a major problem, but the way our food system is set up, it doesn't end up crossing into people all that often in the US. That situation changes in countries lower than the socioeconomic ladder where the food supply isn't so secure. Okay? So the natural hosts for these organisms are various species of grazing livestock, often ruminants such as cattle and sheep. And humans are an accidental host. So go back to walk you through the life cycle then. There are some important differences between these liver flukes and the ones that we just discussed. The adult flukes live in the lumens of the bile ducts just like the other species. So in the intrahepatic portions of the bile duct, of the biliary tree. And they produce eggs that pass down the bile duct to the intestine and leave the body in feces. So that part is the same. And then when the eggs get into the water, they actually hatch in the water. And a larval stage emerges from the egg that then goes off and actively infects the snail. So no need for the snail to ingest the egg. That's not very important. Then infection in the snail proceeds with several rounds of development that you don't need to worry about the details of. And a cicaria emerges from the snail, just like before. But now, instead of infecting a second intermediate host like fish, the metacicaria actually insists on the leaves of various freshwater plants. Okay? And that's actually the ideal place to be, right? If the host that you're targeting next is a grazing animal, an herbivore, it's going to come along and instantly munch on those leaves, score, you've just infected your next host and you've completed your life cycle. Humans become infected when they eat those same plant foods. So there are various species of aquatic plants that are used as vegetables in certain parts of the world, and infection with Fasciola is associated with consumption of those vegetables. Now, there's an interesting difference then that occurs once the metacercaria has been eaten. So if the metacercaria is eaten on these plants, it again passes through the stomach, just like the other liver fluids, reaches the duodenum, where it reactivates and it persists, and the juvenile fluke emerges from the metacercaria. Remember how I told you with the other liver flukes that juvenile fluke finds the bile duct and crawls up the bile duct? That doesn't happen with this species. With Fasciola, the juvenile fluke takes a much more direct and invasive route to the liver. It actually penetrates through the gut wall, directly through the tissues, into the peritoneal cavity, migrates across the peritoneal cavity to the liver, and then pierces the liver capsule and burrows into the liver through the liver tissue until it finds a bile duct that it wants to invade. And then it enters the bile duct and sets up shop and grows into an adult fluke. So there's a much more invasive aspect to the life cycle that actually has potential to cause disease. Okay? There's actual destruction and damage to tissue as these juvenile flukes are migrating through the various tissues. Just a summary of some of those points. I said humans are really an accidental host for this species. Various species of domestic animal are the typical hosts, but also various wild animals can act as hosts. However, there is evidence that this infection can maintain itself in humans. So if the circumstances are right, you don't need involvement of these other hosts in order for the life cycle to be maintained. All right, so some examples of vegetables that can be a source of infection are plants such as watercress. Okay, so if you enjoy your watercress sandwiches, if you eat watercress sandwiches in the US, that's totally fine because our watercress is farmed. Okay, it's not gathered from uncontrolled wild environments where it could become contaminated with feces of Fasciola-infected grazing animals. So farmed watercress in the US is not an issue. It's not a source of infection. But in parts of the world where watercress is gathered from natural locations, and especially from locations close to where livestock may be grazing, this is a potential source of infection. As an example, just one of the snail species that can act as an intermediate host. And then this is the metacercaria, which is tiny, not really visible to the naked eye. You wouldn't notice it if it was present on the leaves of these plants. Some pictures of the adult flukes. So some of these are really impressive looking organisms, over an inch in length. And now with these organisms, the typical presentation again is of a chronic biliary stage due to the presence of the adults in the bile ducts. But because of that invasive nature of the juvenile flukes on their way to the liver, it can also present as an acute hepatic stage where the larvae are migrating through the liver tissue. That can lead to symptoms and over disease. This typically happens only when a large number of metacercaria are eaten all at once, but again, it can happen. And this can last, as it usually occurs, several weeks after ingestion of the metacercaria. It can last for several months as the flukes make their way through the liver tissue to the bile duct. And it's associated with abdominal pain, intermittent fever, malaise, and weight loss. And there's usually a pronounced eosinophilia in response to this infection, as there is with most parasitic worm infections. After this, it can progress to this chronic biliary stage where obstruction of the bile duct can occur. So the flukes are now in this luminal location, and because of their large size, and if sufficient numbers are present, then there can be intermittent obstruction to the flow of bile through the bile ducts. This leads to abdominal pain, can predispose to ascending cholangitis, bacterial infection tracking up from the intestine, and can lead to jaundice and the formation of gallstones. Some pictures of what this looks like. These are actually images from a slaughterhouse in the US. This is a cow's liver over here that's been cut open. You can see the fibrosis of the bile duct wall over here, and then all of these flukes jammed into the lumen of this bile duct. One of them has been flipped down onto the cut surface so that you can see it more clearly. This happens to be a sheep liver over here that's been cut open. You can see this tremendous fibrotic reaction occurring around these bile ducts. An image, a histological image here of a fluke in cross-section with fibrosis surrounding the bile duct. Again, fibrosis, because of the chronic nature of this inflammation, is a major feature of the pathology of these infections. Diagnosis, again, usually by demonstrating the presence of eggs and stool. There are various serological assays that can be done to detect antibodies that the body is producing against Fasciola antigens. I just mentioned this potential for the diagnosis of false fasciolitis, a pseudo-fasciolitis, and we see fluke eggs in feces of the individual. It's worth exploring the option as to whether or not they may have eaten liver as a food source. Okay? So if the liver of an infected cow or sheep was eaten, it's possible for the eggs to pass straight through the GI tract and be present in feces, leading to this false diagnosis. Just a word of caution. So the eggs of Fasciola are actually some of the biggest eggs you ever find in human feces. These are gigantic, up to 150 microns in length. Again, we'll have some of these for you to look at this afternoon. They also have an operculum. You can see it's popped open on this egg here. This little scape patch that allows the myristidium inside to emerge. Now, unfortunately, there are several different species of fluke that have eggs that look like this, and so it usually takes considerable expertise to differentiate Fasciola eggs from those of some of the other species. The treatment of Fasciola, so this is the one flatworm infection that you cannot treat with Praziquantel. Okay, this is the big exception that I hinted at earlier. The drug of Praziquantel does not work against Fasciola. So the drug of choice for treating Fasciola infections is actually this drug, triclobendazole. Okay, so in the last few minutes of this hour then, we'll quickly run through this last genus, Paragonmus, the lung fluke. Are there any questions about liver flukes before we move on? Yes, sir?
I didn't quite understand human to human.
Oh, yeah, so I should have worded it human, snail, human. Yeah, there's no, with all of these flukes, the intermediate host is actually essential for the completion of the life cycle. So transmission human to human in any of these organisms. Apologize for not making that clear. Another question?
The same destructive pathway you mentioned with the humans is, exits through the gut, goes to the perineum, and that goes to the liver caps. Is that the same in the sheep as well?
Yes, that's absolutely right.
Same process.
Yeah, exactly. And so my background is I'm a veterinarian, so I'm familiar with seeing that presentation in sheep and cattle. Yes.
So if the human eat metacerca from the pork or from the cow, can the transmission?
I'm sorry, I missed the first part.
If the human eat metacerca, what is that? The metacercasis from the pork or from the cow, meat or liver,
Yeah,
can be
okay.
transmission?
Yeah, no, so you can't get infected by that route either. The question is whether ingestion of infected cow liver or infected sheep liver actually results in infection. No. The life cycle has to go through that snail intermediate host and produce a metasacharia. It's only the metasacharia that's infectious. So ingesting infected cow liver or sheep liver sounds kind of gross, but it's not actually dangerous. It's just good invertebrate protein, if you like. So there's good food protein in your diet. Yes, sir.
From a health point of view, is it